1,000 kilometers. To survive a flight this distance, an animal must cross mountain ranges, navigate shifting weather fronts, and outrun aerial predators without stopping. For a bird weighing less than a pound, maintaining flight for 14 consecutive hours requires a metabolic efficiency that pushes the absolute limits of avian physiology. Every gram of fat must be perfectly converted to kinetic energy. The animals capable of this are not wild. They are engineered by a specialized community of breeders who isolate specific survival traits over generations. The financial stakes are massive. A single bird with elite marathon genetics can command hundreds of thousands of dollars on the global market, subjecting these animals to intense, data-driven scrutiny. You cannot train a pigeon to fly 1,000 kilometers if it lacks the biological hardware. Winning at this extreme distance is entirely a problem of genetic engineering. In the high-stakes world of extreme long-distance racing, the Belgian lineage cultivated by Etienne Meirlaen is recognized globally as the apex of endurance genetics. This is a pedigree chart from the Meirlaen loft. For decades, dense, text-heavy documents exactly like this have been the primary tool breeders use to design their champions. In practice, this document functions as a directed acyclic graph, a mathematical map used to track how specific biological traits are concentrated over time. The final output of this specific graph sits on the far left, a modern racing pigeon identified as B214039-257M. By reverse engineering the historical data plotted across these nodes, we can uncover the exact mathematical and biological strategies required to fix extreme endurance directly into a population's DNA. To understand the final product, we have to look at the starting genetic material. That requires moving backward in time, to the ancestors on the right side of the graph. Each rectangular node represents an individual ancestor. Here, breeders record quantifiable data to prove the bird's genetic worth to future buyers. The text inside this specific node for Montali serves as a rigid performance dataset. It lists top national placements in the grueling KBDB races, showing Montali placed 4th against a field of over 9,000 birds from Montauban. Biologists call this phenotype, the observable physical expression of an organism's genes. A 4th-place finish in a marathon race is proof that Montali possessed elite endurance capability. Breeders employ strict visual management systems to track these proven phenotypic traits down through the generations. The color coding acts as a visual sorting mechanism. The red and pink boxes trace the stamina-heavy Yelena bloodline, distinct from the Marseille lineage marked in green. In these older generations, this historical race data provided the only available proof of genetic worth. A bird's physical survival on race day served as the primary verification of its internal quality. But breeding for extreme traits introduces a biological paradox. If you make closely related champions to concentrate their endurance, you trigger inbreeding depression, a compounding of mutations that ruins the immune system and weakens the animal. The solution is a precise geometric strategy called line breeding. Breeders mate animals that are related, like half-siblings or cousins, but spaced just far enough apart in the family tree to safely lock in desired alleles. Visually, line breeding manifests on a graph as deliberate points of convergence, where distinct ancestral branches fold back in on themselves. We can see the physical evidence of this strategy directly on the primary Meirlaen chart. Notice the foundational Yelena node. It is deliberately engineered to reappear multiple times within the paternal top half of the lineage. By crossing half-siblings who share Yelena as a common ancestor, the breeder mathematically doubles the probability her specific endurance genes will pass to the final offspring. The second pedigree from the same loft, charting a completely different modern bird, confirms that this concentration technique is a systemic methodology. Here, the graph relies on the exact same structural convergence, but this time it isolates and preserves the green Marseille lineage. The DAG structure allows breeders to mathematically maximize the frequency of winning genes, while maintaining just enough distance to suppress the homozygosity of fatal mutations. Because this biology is successfully fixed, it functions as an exportable technology. These genetics actively propagate thousands of miles away from Belgium. In the Philippines, breeders like Kirby Chua acquire these precise European lineages to compete in the notoriously harsh tropical racing environment. Looking at this modern Philippine pedigree certificate, we see how the imported Belgian genetics have been seamlessly integrated. Even halfway across the world, the graph relies on the same foundational architecture. The Montali and Yelena nodes reappear on the right flank. But there is a critical evolution in this modern document, a data point that was completely absent from the historical Belgian charts. As we move toward the modern offspring, the data recorded inside the ancestral nodes shifts from physical race results to direct molecular chemistry. The breeder is now directly testing for and verifying the LDHA genotype. LDHA stands for lactate dehydrogenase A. It's the gene responsible for regulating how avian muscle cells process and clear lactate during intense, sustained physical exertion. Possessing the rare AA variant of this gene accelerates lactate clearance. Without it, a bird attempting a 1,000-kilometer flight will suffer systemic muscle failure and simply drop out of the sky. Including this gene in the chart marks a transition from relying on statistical racing results to possessing molecular certainty. The breeding industry has evolved from observing external performance traits to mapping the internal chemistry of the animal. The elite racing pigeon is a meticulously constructed biological machine designed to execute a singular, brutal task. When we pull back and view the pedigree as a global data structure, we see a vast network spanning decades and continents. And the modern DNA tests verify what the historical phenotypes suggested. The line breeding strategies successfully forced the endurance genes to the surface. These pedigree charts function as precise, living blueprints, mapping the physical concentration of endurance across space and time.